Okay.
Okay, so the plan is we're going to finish up the first part of Neoplasia, and then we're going to take a look at the questions. There are a subset of questions we can already do, and I'd rather get to them while the information is fresh, and then we'll finish the second part of Neoplasia and do the second part of the question. So, last time we discussed some of the specific differences between benign and malignant neoplasms. So you should get an idea by now of just how these different types of neoplasms will present. You should be able, for example, to recognize the difference between a benign and malignant neoplasm just by inspecting the gross appearance of the lesion. Benign neoplasms usually have a smooth, well-delimited border, well-circumscribed, whereas malignant, usually irregular, blurry kind of borders. Okay? We talked about those different characteristics. We talked about how some characteristics correlate with benignancy and malignancy, while others define benignancy versus malignancy. Okay, so that you should be comfortable with right now. Okay, we're going to take a look at some other things. First, we're going to take a look at some epidemiologic correlates. Okay, these numbers are a little bit dated, but certainly there are over 1 million cases of cancer that are newly diagnosed each year. A couple of important factors that increase the prevalence of cancer. Age, for example. The frequency of cancer increases with age. Basically, the passage of each decade of life increases the risk of cancer. There are certainly good reasons for this. Number one, cancers usually develop as a result of an accumulation of mutations in the cell's genome. As we age, cells replicate, they make mistakes in their DNA replication. Those mistakes accumulate as mutations. The DNA is damaged over time, and so as we age, more and more of our DNA gets damaged. And the more of our DNA gets damaged, the greater the probability a cell will lose proliferative control. So that certainly makes sense. But there is another important reason why the incidence of cancer increases as a function of age. Our immune systems become less competent as we age. It's thought that a lot of different cancers develop in our bodies and we never know about it. And that's because our immune systems basically recognize the development of these cancers and destroy them before they become clinically significant. However, the immunosurveillance of our immune system declines as we age, and there's a greater opportunity for cancers to develop, and there's less of a chance your immune system will recognize and destroy it before it becomes clinically important. And so those are the two major factors that seem to correlate with the increase in cancer as a function of age. Altogether, most mortality occurs between the ages of 55 and 75. It seems that if someone's going to die of cancer, it's going to be somewhere in that age range. Cancer is also responsible for about 1 in 10 childhood deaths. Usually, childhood cancers are sarcomas, some type of sarcoma, and that's because when the child is growing, it's the sarcomatous, it's the connective tissue that is developing in these children. That's the part that's growing, and so it's more likely that as this tissue develops, something's going to go wrong. And usually, connective tissue being derived from mesenchyme results in some sort of sarcoma. Excuse me. So, usually when a child develops some sort of cancer, it's usually a sarcoma. Now, when a child develops cancer, usually it's not because the child has acquired mutations in the DNA. It's usually because they inherited mutations in their DNA. They develop some sort of inherited paraneoplastic syndrome, some sort of neoplastic syndrome that predisposes them to develop various types of cancers. Typical cancers, again, sarcomas, bone sarcomas, things like lymphomas and leukemias. lymphomas used to be called lymphosarcomas. Okay, again, these are types of cancers associated with mesenchyme, the type of tissue that would be growing in young children. All right, any questions on that? Okay, let's have a look at some of the different types of cancers that tend to kill people. We'll take a look at the frequency of cancer associated by death and sight. Okay? Here's a nice little chart showing you the different types of cancers responsible for deaths. These are in various proportions. These are not the most frequent types of cancers, but these are the frequency, showing you the frequency of cancers associated, frequency of cancer-associated death by sight. Okay? These are the types of cancers that tend to kill people. In both men and women, the most common type of cancer that ultimately results in a patient's demise is lung cancer, accounting for 32% of cancer-related deaths in men and 25% of cancer-related deaths in women. And presumably that's due to cigarette smoking. Okay? So that's not really a surprise. The next most common cause of death in men is prostate cancer, accounting for 14%. Okay? And in women, it's breast cancer, accounting for 17% of cancer-related deaths. Now, these two tissues have something in common. Both these tissues are hormonally responsive, meaning that they will grow and involute in response to fluctuations in hormones. And anytime you have a tissue that's growing and involuting, you know that tissue is constantly replicating cells and DNA within those cells. You replicate the DNA, you don't do so with 100% fidelity, and mistakes are made, and some of those mistakes persist as mutations. So any tissue that's associated with growth and involution is more likely to be susceptible to developing cancer, and that's what happens here in the prostate and in the breast tissue. Now, the breast tissue is responsive to hormonal fluctuations that occur during the menstrual cycle. Mostly estrogen and progesterone are influencing the growth of the breast. Estrogen levels increase during the menstrual cycle, and the breast develops. Some of the cells become hyperplastic. While at the end of the menstrual cycle, well, the cells tend to involute via apoptosis. So it's constant process by which the cells are growing and they're involuting during the menstrual cycle as a result of hormonal fluctuations. Okay? Not surprising then that breast cancer is one of the more frequent cancers that cause death in women. Now, prostate also is responsive to hormones. Men also go through cycles of hormonal fluctuations. The cycle is not as well defined as that of the menstrual cycle of women, but men do go through these different cycles of hormone fluctuations. And the prostate, therefore, goes through the same process. It grows and involutes, and all of the cells in that prostate are going to have to replicate their DNA, they're going to make mistakes, and again, you get the degeneration of some of the genetic structure, the genetic composition, and that predisposes to cancer. So prostate cancer is pretty common in men. In men 80 years or older, about half of men have prostate cancer. We know that because when we perform autopsies on men in that age range, we discovered that half of the prostates contain cancer. Now, that cancer may not have been clinically symptomatic, but we know that about half of prostate glands, again, older, in men older than the age of 80, are remarkable for prostate cancer. Whether prostate cancer is symptomatic or occult doesn't matter. Half of men over the age of 80 have prostate cancer. Okay? So it's a common type of cancer, but it only accounts for about 14% of deaths caused by cancer. Okay. Any questions on this? All right, let's take a look at some different types of hereditary syndromes associated with the development of cancer. And there are a number of genetic mutations that one could inherit that will predispose them to developing cancer. There are some common types of cancer, often caused by these mutations. A good example would be retinoblastoma. Retinoblastoma is a nasty cancer of the retina. Very primitive type of cancer. The more primitive the cancer is, the more aggressive it tends to be. Well, retinoblastoma is kind of a nasty cancer of the eye. Usually develops in kids. And it's because these kids have inherited a mutation in a gene that normally suppresses the growth of cells. When this gene is mutated, you get a dysregulation of cell growth, which is the basis of neoplasia. The gene that's mutated, that these kids inherit, is called the RB gene. RB stands for retinoblastoma. It encodes for a gene that normally suppresses cell growth. It's called a cancer suppressor gene. And when one of the genes, either the gene coming from mom or dad, is mutated, the person receiving the mutated gene has a 10,000-fold increase in the likelihood of developing this type of cancer of the eye called retinoblastoma. And we're going to talk more about this gene. And it is a cancer suppressor gene. And usually you get one gene from your mom, one gene from your dad. If either one of these genes, either from mom or dad, is mutated, you have a 10,000-fold increase in the risk of developing retinoblastoma. Now, usually it's the parents that discover the retinoblastoma. Retinoblastoma is a bulky kind of cancer. It grows in the back of the eye. And usually the parents notice something wrong about the child's eye when they're taking a photograph. And when taking a picture of a child, usually with the flash you get this red eye reflex, called this reflection of light in the back of the eye. It's called the red eye reflex. Okay? That's my cat, red eye reflex. Okay? You should be able to see that red eye reflex. However, when a child has a cancer growing in the eye, the retina, the back of the eye is blocked. And so when they take a picture, instead of seeing the back of the eye, the red reflection, they see the white reflection of the surface of the cancer. And this is what it looks like. Here's a child with an abnormal red eye reflex. Okay, it looks like the right eye is okay, but the left eye you can see is white. That's called leukocoria. White. Leukocoria. The person's eye here is filled with just a lot of cancer. It's blocking the back of the eye, and as a result, instead of seeing that red eye reflection from the back of the retina, you see just the surface of the cancer, which is white. And this might be the first tip-off that something's wrong with the eye. Okay? This is what an advanced case of retinoblastoma looks like. Okay? You have a depiction here of the mass growing in the back of the eye. Again, the mass can fill the globe of the eye. Okay? This is what it looks like when it starts to invade the cornea, goes right through the pupil, gets into the cornea, and starts invading the cornea. Now, the cure for cancer, if the cancer remains localized and you can resect all the cancer cells, you can achieve a cure. Usually when we resect all of the cancer cells, we perform something called a wide area excision, meaning we take out tissue. Not only do we take out the cancer, but we take a wide area of normal tissue as well. That's called wide area excision. Okay, we want to guarantee that we're capturing all the cancer cells. If you can do that, you can achieve a cure. Resect all the cancer cells, usually you can achieve a cure. To perform a wide area excision on this case, you'd have to enucleate the eye. You'd have to remove the eye. It's really the only way of curing it. If the cancer if the cancer remains localized, you do that wide area excision. You should be good to go. person obviously loses the eye, but if it is not removed and the cancer remains, it's going to metastasize it's going to metastasize all over the place, and it's going to invade the brain. Any questions on that? Okay, that's an example of a hereditary form of cancer. About 40% of retinoblastomas are somehow familial, they're somehow genetically related. The remaining types of retinoblastomas develop spontaneously, but clearly a large percentage of these retinoblastomas develop as a result of inheritance. Yes? What's the percentage of cancers that are hereditary? That might be a hard question for me to answer, because some people are more resistant to cancer than others just by genetics. So depending on how you're defining that, I would, if I go with a kind of a loose definition, which cancers are kind of acquired based on acquired condition. My experience in adults, it's got to be more than 90%. It's not very common. Most people are going to be diagnosed in childhood with cancer. And acquired types of cancer in kids is almost unheard of. They have to go through some process of mutations that takes decades. Okay. All right, so let's take a look at some other types of inherited cancers. We're going to take a look at something called adenomatous polyposis coli. This is a condition in which there is a mutation, again, in a gene that regulates cell growth. This mutation usually initially presents with the formation of thousands of polyps in the colon. That's why we call it adenomatous polyposis coli. Adenomatous polyposis coli. It first presents with a lot of different polyps in the colon. Now, someone with adenomatous polyposis coli initially presents with these polyps. However, these polyps are invested by a dysplastic epithelium. And you know that dysplasia is the forerunner of cancer. And so ultimately what happens with one of these polyps, that dysplastic epithelium degenerates into a bowel cancer. And so someone with adenomatous polyposis coli has virtually 100% chance of developing colon cancer by the time they're 50. It's going to happen. Let's take a look at this. Let's first take a look at the normal colon. Here's a resection of terminal ileum, cecum, and ascending bowel. There's the large bowel right there, as you would see it after it's been opened along its longitudinal axis, and you're looking down at the mucosal surface. You have lots of folds here. Okay, everything looks good in this part of the colon. It's a normal specimen. It's kind of got that yellow color. That's from bile coming from the gallbladder, liver. It kind of stains the surface that color. Everything looks good there for the most part, at least in the ascending bowel over here. Everything's normal. If that's what normal colon looks like, here's what adenomatous polyposis coli looks like. That's a resected large bowel. Looking down at the mucosal surface, and you can see there are literally thousands of polyps here. Now, and you consider each one of those polyps is infested by a dysplastic epithelium. What are the chances that the dysplastic epithelium of one of these polyps is going to degenerate into cancer? Like I said, it's virtually 100% by age 50. So someone with this condition, they usually opt for a prophylactic colectomy. They want to have their colon removed before the cancer develops. If you do that, then there's very little chance that a person will develop bowel cancer, okay, which is almost a certainty by age 50. So they opt for an elective prophylactic colectomy. Okay. Let's take a look at why this condition is such a nuisance. We're going to take a section through a polyp. Okay. Here's the polyp. This polyp has a growth most morphology we refer to as pedunculated. It's got a stalk and it's got a head. Let's take a look at this. Section through the polyp right there. You can see the lumen of the colon out here. There's some normal colonic wall. You can see there's the mucosa. That's the submucosa right there, and that's the muscularis propria. Everything about this part of the colon wall looks good. Everything looks normal. Over to the right here, you can see where there's a little stalk arising from the wall of the colon, and you can see a head on top of it. You got a stalk, you got a head. That morphology is referred to as pedunculated. This is a polyp pedunculated morphology. Okay. Stalk is invested usually by a normal colonic epithelium. Okay, everything is fine here. The core of the stalk consists of fibrous tissue, vascular tissue, so it's a fibrovascular core. The problem is up here with the head. The head is usually invested by a dysplastic epithelium. Take a closer look at that, zoom in here. Here's a close-up of the epithelium. We've got two types of epithelium here. One is normal, one is clearly dysplastic. It's one of these little villus-like structures. There's the colonic lumen. You've got the epithelium here. That's the lamina propria. Let's take a look at the different epithelial cells. This side of the structure is invested by normal epithelial cells. You've noticed they're columnar type cells. These are the typical enterocytes you find in the lower GI tract. Over on the other side, you've got a dysplastic epithelium. Take a look at the normal epithelium first. Again, you've got these well-spaced columnar cells. This section's a little bit thick, so it looks like the cells are overlapping, but they're not. They're actually well separated here. They all look the same. Got a little microvillus brush border up here. There's a basement membrane that they sit on, right there. It separates from the lamina propria. Everything looks good. Compare that to the dysplastic epithelium. Look at the cells. See how they're crowding each other? I mean, they're climbing all over each other. They shouldn't do that. Clearly something is wrong with the regulation of proliferation of these cells. They're over-proliferating. That's the definition of a neoplasm. All of these cells are neoplastic. Now, take a look at the nuclei. You can see they're clearly overlapping each other. All the nuclei are very different from each other. They don't look the same, not like they do in these normal colonic cells. Nuclei pretty much look the same. All these nuclei look pretty different. That's called pleomorphism. And that is a consequence of the different mutations that they're all developing. They look very different from each other. Now, these cells are proliferating out of control. They're crowding each other. They are pleomorphic. But notice they're respecting a basement membrane that runs underneath here. That basement membrane is a natural tissue barrier. And the cells are not penetrating that natural tissue barrier. So this neoplasm is a benign or malignant. It's benign. There's no evidence of local invasion, which would be a defining feature of malignancy. These are all benign cells respecting that basement membrane. Okay, so all of the neoplasm, but it's not cancer yet. That's a good thing, obviously. Okay, we can also refer to this epithelium as a dysplastic epithelium because it's got all the features of dysplasia, overcrowdedness, pleomorphism, okay, architectural disorganization. That's all in the definition of dysplasia. Okay, everything looks like it's consistent with dysplasia. Now, again, a couple of things can happen with this dysplastic epithelium. It can stay that way. It can remain as a dysplastic epithelium, never give the person in the problem. But more likely, somewhere in the colon, these cells are going to continue to mutate until they turn on an enzyme that allows them to break through that basement membrane. When they do, they're going to invade the lamina propria. When that happens, that's not dysplasia anymore. That is bowel cancer. So, not too difficult to understand why someone with adenomatous polyposis coli who has thousands of dysplastic polyps in their colon, by the time they're age 50, 100% chance of colon cancer. They want their bowel out. They don't want to take the chance, which is almost inevitable. Okay, questions on that? All right, some other inherited syndromes. Xeroderma pigmentosum. Have we ever talked about that? Xeroderma pigmentosum. Do you know what the name Xeroderma pigmentosum means? Can you use Latin or something to figure it out? Greek and Latin. You know what Xero means? What, Xerox? Xerostomia? Never heard it? It means dry. 0 gram means dry. Derma, of course, skin. Pigmentosum, pigmented. So this is dry pigmented skin. That's what this disease is. And this is actually a disease caused by some sort of mutation in a gene that encodes for one or more of the DNA repair enzymes. Okay, there's a series of DNA repair enzymes your body uses to repair DNA that's routinely damaged by, say, UV radiation. Every time you go out in the sun, your skin is exposed to UV radiation, your DNA is damaged. It's common. We have a series of enzymes that work in conjunction with each other to repair that damage. You've got one enzyme that recognizes the damage, another enzyme that comes over and cuts out the damage, another enzyme comes over and rewrites the DNA. Those are collectively referred to as DNA repair enzymes. Sometimes a person will inherit 2 mutant alleles of that encode for these DNA repair enzymes. One comes from mom, one comes from dad. You inherit 2 mutant genes that encode for these DNA repair enzymes. The enzyme doesn't work. And all of these enzymes have to work in series, like links in a chain, in order to repair the DNA. You have one of these enzymes that doesn't work, you can't repair the DNA damage. And you develop a disease called Xeroderma pigmentosum. Xeroderma pigmentosum is caused by one of these enzymes that's normally involved in DNA repair. Now, because you can't repair DNA when it's damaged by UV radiation, young children start developing skin cancers very early on. All kinds of skin cancers. Squamous cell carcinoma, basal cell carcinoma, malignant melanoma. All of these different types of cancers develop very early in these children because they're incapable of repairing their own DNA. We call this condition Xeroderma pigmentosum. Here's an example of a type of cancer developing in someone. They're exposed to the sun. UV radiation damaged DNA can't be repaired. The cells proliferate out of control. You get squamous cell carcinoma. Here is an example of some exposed region of the body, the hand. You can see the fingers here. You can see there's a nasty kind of pus-festering ulcerative lesion there on the finger. That's squamous cell carcinoma, appearing as a kind of ratty ulcerative lesion. Cancer cells have lost proliferative control. They're eating their way through the soft tissue, and they'll eat right through the bone. And this is a result of sun exposure in a person who can't repair damage done by UV radiation. The only way to cure something like this would be amputation of the finger. You have to, again, resect all the cancer cells to achieve a cure. Now, most of the time, these children that have Xeroderma pigmentosum, you wouldn't let them out in the sun anyway. It's almost a death sentence to do that. We keep them generally in the dark. And sometimes they're called children of the dark for that reason. Even UV radiation produced by fluorescent lighting is enough to cause cancers in these kids. Fluorescent lighting damages our DNA. It doesn't do it at a high rate, but it still damages our DNA, so we generally keep these kids in the dark and try to manage them, at least in part, that way. Okay. So Xeroderma pigmentosum, again, another example of an inherited Neoplasia syndrome, this time caused by a mutation in the genes encoding for one or more of the DNA repair enzymes. Now, just one aside. The frequency of the mutant allele in the general population is pretty low. Most people do not carry this mutant gene that they can pass on to their kids. In frequency, really low. The chances that two parents having this mutation that they can donate to their kids is astronomically low compared by chance alone. It's very, very low indeed. And so when a child is born with xeroderma pigmentosa, you might want to consider whether the parents are actually related. They have some of the same genetic material. If they're related, then the chances of both of them having a mutation that they can contribute to the child to develop this disease is not so unusual. When the child is produced by two parents, you first suspect that maybe the parental relationship there is consanguineous, or maybe the relationship between the wife and husband is consanguineous. It doesn't have to be, but that's certainly one possibility. Maybe they're related. They have the same genetic material they're donating to the child. It's also quite possible they can be complete strangers, not related at all, but belong to what we call a genetic isolate, where you have, for example, a isolated population of individuals that intermarry. Maybe the two individuals are not related to each other, but because of the isolation of the population, maybe a lot of the population shares the same genetic material. That could have developed over years of isolation. Good examples, islands. People that live on islands, they tend to all kind of interbreed. And that might be a good example of a genetic isolate. Sometimes social constructs, religious constructs are genetic isolates. Certain types of religions only marry within that religion. That can be a genetic isolate. So something's going on. The two individuals most likely share genetic material. Whether the relationship between the parents is consanguineous or not, well, you'd suspect that something about is related somehow. Okay, questions on that? Next thing we're going to talk about is some of the acquired pre-neoplastic disorders. So we said that some cancers, very few of them, are actually the result of inherited mutations. Retinoblastoma, adenomatous polyposis coli, bowel cancer, and of course skin cancers caused by xeroderma pigmentosa. Those make up a minority, a very small minority of cancers that we see in human people. Most of the cancers that we see are the result of acquired mutations, and cancers that develop as a result of acquired mutations are called acquired pre-neoplastic disorders. Any condition that requires constant regeneration of tissue is a pre-neoplastic disorder. The good example I give you would be liver cancer. Liver cancer commonly develops in people who are alcoholics. Why? Alcoholism destroys the hepatocytes. The liver has to constantly regenerate. And as a result, they replicate their DNA. They make mistakes. As a result, you get a neoplasm developing in the liver. Hepatocellular carcinomas. Not common in the general population, but very common in alcoholics. The example of a pre-neoplastic syndrome. If you have a wound that is non-healing, for example, chronic osteomyelitis, which is an infection of the bone, very commonly seen somewhere on the leg, bone gets infected, the pus in the bone eventually drains through a fistula on the surface of the skin. The skin in the vicinity of the fistula constantly degenerates and has to regenerate. We very often see squamous cell carcinoma developing in the vicinity of the draining sinus. That's another example. Any kind of dysplasia or hyperplasia. Remember we talked about atypical hyperplasia of the endometrium, usually driven by high levels of estrogen. glands overproliferate the endometrium. Woman presents with heavy bleeding, at least initially, and that's about it. That's a risk factor for endometrial cancer. Endometrium's constantly proliferating, it's overproliferating. Again, cells are replicating at an unusual rate. They're making mistakes when they replicate their DNA. So, atypical endometrial hyperplasia is a risk factor for endometrial cancer. Okay, any questions on that? Why don't we take a break and then we'll take a look at environmental related cancers, occupation related cancers.
I'm gonna we just sit at this desk, put the game on, and be like, okay, game on. No, I can't watch the game. I can't watch it. That's
Wait,
where it's like,
I didn't hear you say that.
and that's where it's self.
They make their own seniors.
And that is you also lose the
What's the shorter? You can say what you want, but I think that's just an excuse to take a shot. That's straight fuck. Well, I can like crush weakness.
Exactly.
Can go below.
There you go. Ready? I remember everything.
Oh yeah.
Ever since one, just one.
Hey.
Ever since you said swamp water last night, that's when I wanted to. Okay.
No,
Just
you
one.
literally get it.
so you know.
Go for
I
it.
have a better memory juice. Actually, I have
Maybe I didn't.
You got some too. Oh. What did you get? What did you get?
Sorry. Yeah.
So many.
I thought we had a screen
That's like
touch bar.
it's cool.
So is that like an A or Pro?
Your screen is so tiny. That's crazy. I
That was-
think it's really big.
That was the cheapest one.
Yeah.
I think it was $7. So like I'm only
It's 10. Okay. We're going to have a fire alarm, I guess, simulation 11. So, by the time we get back, probably going to a no-go for class. We won't have much time left. So, we can pack it up now and don't forget we're meeting tonight. Finish this up. And,
Do, 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 do.